I'll just start things off, Megan, if you don't mind.
Yeah, go for it.
Yeah, awesome. Well, welcome everybody. I'm happy to see so many people here training to be a part of the Macrophyte Survey Program. My name is Sam Beck-Anderson. I am the Aquatic Invasive Species Program Manager at the Finger Lakes Institute and Finger Lakes PRISM. The Finger Lakes Institute, for those of you who don't know, is a interdisciplinary non-for-profit department at the Hobart and William Smith colleges. So we do all kinds of different research and outreach, education, programming for a number of different projects all around the Finger Lakes region. We have research programs going on from, focusing on anything from harmful algal blooms to mercury bioaccumulation, and of course, a large amount of invasive species programming as well. And all of these programs essentially converge on, the common theme of the health and fortitude of the Finger Lakes watershed here in New York. So with that, we're really excited about this program. I'll let Megan do most of all of the talking. I'm going to be signing off here in a little bit. So I will introduce here Megan Harris to take over the rest of the day here. So Megan, good luck and glad to see everybody here. And yes, we'll be in touch soon.
Cool. Thanks, Sam. Have a good day.
Yep. Thanks. Take care.
All right, so I'm just gonna hop into it. I'm gonna share my screen. Oh wait, Sam.
Yes.
Can you? I'm not able to share my screen.
Okay. Good thing. Okay. Let's see. Participants. Need to find you on the list. There's Megan Harris. Make host.
Awesome. Perfect.
All right. And let me know if there are any other issues. All right, Megan?
Yep, will do.
All right.
Thanks.
Thanks. Take care. See ya.
All right, here we go. Oops. All right, so this is the Macrophyte Survey Program, engaging community members to prevent the spread and impacts of invasive species on our natural resources. This is a picture of the rakes that we use. I've also got one right here that might be, I can't actually see myself. Here we go. So this is a better version. That one's kind of only got the one side, so we've had problems with it in the past, but yep, just a rake with the top cut off and zip ties right here and here. Yeah, we just throw them into the water and it picks up plants. And these are some samples on the left or on the right hand side that people have sent in past programs. Got some Eurasian water milfoil in there. All right, so what are we going to be talking about today? I'm just going to give you a quick overview of the Finger Lakes PRISM, about invasive species and their impacts. I think those are actually reversed. A little bit about the MSP itself. Starry Stonework Collaborative is part of about prisms. And then the most important thing is how to collect our data. We've got a Facebook group so that you can put out questions to the entire Macrophyte Survey Program, and aquatic invasive species identification. So what is an invasive species? It is non-native to the ecosystem under consideration whose introduction causes or is likely to cause economic or environmental harm to human health. So just overall, a bunch of negative effects. Economic would be some Hydrilla has been known to invade machinery, some certain businesses that use take water for cooling. It can clog up those kind of, that kind of equipment. Economic also would be just the huge impact on tourism. So in this top right photo is water chestnut in all its glory. That looks like you could walk right across that mat of water chestnut. It doesn't even look like there's any water there. People are definitely not trying to swim through that. And fish aren't hanging out there either, that's for sure. And this is our most notorious species, Hydrilla. You can see that there's like a couple instances of pondweed, it looks like right there. Maybe some, nope, I'm pretty sure that's Hydrilla coming up. And it's just sprouting. It's got the bottom covered, so now it's just sprouting up. And it really just takes over any of the native species. Aquatic invertebrates and fish don't feed or breed in there. They just don't interact with it overall. So that would be the environmental aspects of it. Also, this water chestnut over here, is taking over the waterway. There's no way sunlight is penetrating under there, so nothing else can grow, even if it wanted to. And no other plants are there to produce any dissolved oxygen in the water. And then when these guys die, it's an annual plant, so they'll die at the end of the season. And that uses up, the decomposition uses up a lot of dissolved oxygen, and just overall bad effects for the ecosystem. Also, human health impacts, for example, curly leaf pondweed is a huge, a lot of people expect that it causes harmful algal blooms because of it when it's decomposition releases nutrients into the water column. And there are multiple, people are still doing a lot of research actually in the FLI itself. We've got a lot of grad students looking into harmful algal blooms and trying to figure out what the actual effects are. Invasive species they're all pretty similar in the ways that they all use at least some of these aspects to take over the area where they've been introduced to. They're able to survive in harsh conditions. They have no natural enemies. So for example, water chestnut, waterfowl won't feed on it or interact with it. There's no control of it from that aspect. And curly leaf pondweed, one of the invasives, it's one of the earliest species to start growing during the season, so it gets a head start on everything else and can really take over the waterway. Quick growing, outcompetes native species for habitat, and they have high rates of reproduction. So water chestnut can go from 1 plant to 100 plants the next year. It doesn't, just one rosette isn't the one plant. It's actually got, that one plant will put off shoots for a bunch of other rosettes, and each rosette can hold about 10 seeds. So it adds up really quickly, especially when you've got a giant mat like that one we just saw. I spoke about how it's got no nutritional value, reduces or degrades habitat for food and native organisms, second largest threat to biodiversity, and is leading source of environmental and economic damage across New York State. They're spread in a variety of ways. You can see this little picture down here. I thought it was funny that they put a laptop, but the laptop's not spreading the information, but internet sales of species that aren't supposed to be sold, people wanting to use them for ornamentals to make things look a little prettier. Some of them are pretty, but the impacts for a minute, but then the impacts when you lose everything else is when you notice it. Pet trade and aquarium trade. Hydrilla is actually believed to have been introduced as someone emptied their aquarium into the water, and those guys can just sprout from any piece of plant material. So that's how it got established, most likely in the Finger Lakes. Ballast water, that's mostly going to be for plant fragments or like zebra mussels. They're larvae called villagers. They can grow in the ballast water, and then it'll be released into the next water body, and then they can just hitchhike on a variety of products. We've got some forest insects that are really bad, like the spotted lanternfly, emerald ash borer. Those guys can hang out on firewood if it gets transferred. So there's just plenty of ways that invasive species can be spread. One of the biggest things for water chestnut would be there's a lot of concern in Cross Lake over water chestnut, and some of the landowners are just snipping it and letting it float down the river, which spreads those seeds way farther than they normally would, because they actually have these pretty thick nutlets that wouldn't transfer so far normally. So how do we go about managing them? This is the invasion curve. Any land manager or species, anyone trying to control any invasive species knows about this. So your biggest chances are by prevention. Like I said, a lot of these guys will produce seeds at alarming rates. So once they are able to grow for a couple of full seasons, then they just spread really fast. So once they start getting here, we might just try to contain them. And then once it's up here, it's going to take a lot more. You're going to be needing mechanical tools, especially on water bodies or machineries. They have chemical treatments that occur. So this is why we focus so much on prevention, which is a huge part of the Macrophyte Survey Program, which I'll talk about even more. So like Sam was saying, this is our PRISM. We are Finger Lakes right here. It's state and federally funded. So we've got them throughout the state in different regions. Ours is the largest. There are 17 counties in our region, and we work with a lot of lake associations, and which I'm learning is how a lot of people found out about us, which is awesome. The Macrophyte Survey Program itself, and soil water conservation districts, pretty much anyone who will listen. So the partnership is a huge aspect of our job. And our goal is to reduce the introduction, spread, and impact of invasive species within the Finger Lakes PRISM region through coordinated education, detection, prevention, and control measures. This is an example of one of our programs and how seriously we take that partnership aspect of our mission. We've got the Starry Stonework Collaborative is in partnership with the Great Lakes Restoration Initiative, I believe it's called. And they work together from Canada to Michigan to Wisconsin to New York to try to take care of Starry Stonewort, which you'll see is one of our 3 highest priority species. And this is the expansive Starry Stonewort that they've recorded so far. I believe this is from 2019, so maybe a little out of date, but I did ask the guy who runs the program, David Carr, I'm so sorry, I have no control over these slides, if this was up to date, and he said he believes it is. But you can see we've got a good bit of Starry Stonewort over here in our region, unfortunately. And the PRISM focuses on all invasive species, not just the aquatic ones. This is one of the biggest pests that we are focusing on right now is the spotted lanternfly. It's all over Pennsylvania, and there are only a few instances of it in our region right now, or in New York State. They're not in our region as far as I know. But they feed on trees, multiple different types of trees, just a really wide range, and they secrete this honeydew that breaks down parts of the tree, and overall the feeding just stresses it, and it can kill off an entire tree. These are the different stages. Pretty recognizable, fortunately, so if you see it, definitely report it. It's just an example of giant hogweed. That's considered a public health concern because if you brush up against it, the sun, your skin will have a reaction to the sun, and you can get rashes for years because of it if you don't keep that exposed area out of the sunlight. And we've got a similar program to the Macrophyte Survey Program. You probably saw it if you signed up on the Google form for this program, the Terrestrial Survey, where we have people go out on the trails and look for certain invasives and let us know what they find. So like I said, prevention is the key. That's our best chance. That's why we have, this is our one of our watercraft stewards. We've got about 25 of them this year. We go to launches all over the place, from Canandaigua to Otisco, back to Kyuga. Kyuga is a huge one because we've got Hydrilla there. And we've also put an effort specifically for Hydrilla. This top right corner is an example of a little kit that we gave to people who are most likely to run into it, and so that they can have it on hand so that they can let us know when they find it. So the Macrophyte Survey Program helps by allowing us to have a better view of what's going on in our region, and EDRR stands for Early Detection Rapid Response. I'm going to go through all of these in greater detail. Engages the community and builds statewide database of aquatic invasive species. So the main thing is that it expands our reach. Across these 17 counties, there's no way we could possibly survey every single spot in every single lake, every shoreline. Luckily, invasive species don't grow in every single part of a lake because they have to have some sunlight, so they're cut off at a certain depth. But so that makes it easy to have people sampling from the shore or from their kayaks if they're going from the shore. On your kayaks, you can get to go across more communities. It's pretty fun, for me at least. I hope you guys enjoy it too. So filling gaps in monitoring efforts, and it's a really good way to reach distant counties. We've been able to get some people work with the Soil Water Conservation District out in Cortland and Shenango County. We've also expanded it by including our watercraft stewards. So one of the biggest places that are considered most likely to have a new population of aquatic invasive species established is at boat launches, where things might be hanging onto boats or on the way in or out. And also that also applies for trail surveys, where it's most likely that invasive species are going to be found first on the side of the trail because of just the most human interaction. Certain things like some soil pathogens can spread from the dirt stuck on your shoe. And these are some samples that were sent in last year. European frogbit, water chestnut, that's our high priority, and also Starry Stonewort is a high priority species. And community engagement. I'm really excited to be teaching you guys about this stuff. This is what I want to do. I'm just excited about this job in general, if you haven't gathered that from my emails. But community engagement is a huge part of the goal for the PRISM itself. And we want to let people know, so that people know where they can go for this kind of information. It was really awesome this morning with the 3 volunteers I was with, that being able to see how much it helps to be able to see the plants right there and to be able to be that resource. We also have a field guide that is pretty comprehensive and inclusive of any species that you'll be able to find. So yeah, just overall engagement of the community, spread information to others outside of the program, tell your friends. And then hopefully we'll be able to create some long-lasting connections. Data collection, as I've said, expands our reach throughout the region. We are able to see, we have people report native and invasive species. So this is our chart from last year. And the green indicates percentage of native and the blue is percentage of invasive. So it's pretty satisfying to look at and see that most of them are over 50% green. So we're not doing too bad up here. I definitely think that it's worth the effort in the Finger Lakes to manage the invasive species in our lakes, especially considering how much it provides for the economy, all the lakes in the Finger Lakes. So this is one of the aspects that we measure is the density on your rake. We don't look at this information as much, but it's still, if we ever wanted to, we have it there to take a look, and it could provide information on the plant community in general. If there's if it's just full of plants, like maybe it's a good spot, full of native plants, then maybe it's a good spot for fish to be breeding in. If you're pulling your rake across 5 feet of floor and you're not getting any types of plants, then it's definitely not going to be a spot that's being used as habitat or it's a good spot to hide from predators or breed. So that's some information we can gather with the program. And then we ask people to do their rake tosses in the same spot every time, which I'll go into more later. But that gives us information on the phenology of the species in that area. So you can see the difference last year as we went from July to August to September in the amount of curly leaf pondweed reports. That's curly leaf pondweed, which is a species that starts growing first in the season. Like it's ready to go. Anytime the water temperature starts to rise any amount, it's like, here we go, guys, it's showtime, and starts doing its thing. Hopefully, also we have a lot of people in Tompkins County this year, so hopefully we'll be able to see some of that information with Hydrilla. I think that would be really interesting, because Hydrilla, that's where some of our populations have been in the past. Hopefully we find no Hydrilla, but if we do, at least we'll gain some information on it. So this is the Macrophyte Survey Program as it's been for the last few years. This year we have a lot of volunteers, more than we ever have before. As long as all the people who've responded participate, we'll have at least 100 volunteers across the entire region. So I'm really excited about it. It's started out, there's been a lot of changing over the years, tweaking data. We're still tweaking data. We're trying out IMAP invasives this year, which is really simple and really important because it's a statewide database. But yeah, that's our history in a nutshell. We've depended a lot on soil water conservation districts and lake associations in the past. So I'm really hoping, I'm glad that we've got some who aren't as familiar with the efforts going on in the lake, hopefully this year. But everyone's important, no doubt. Since the start of the program in 2017, volunteers and stewards have done almost 1,000 rake tosses. That's pretty awesome. This is just a little more data about 2020 in specific. We've got some samples down in the corner, and this was our distribution. So last year was pretty successful. Got 27 water bodies, 430 rake tosses, and this is that same map that I showed you before. So how does it work? You gotta attend the training, which you're doing right now. Great job, everybody. Take samples once every 2 weeks, or once in every 2-week period, and report your findings using IMAP invasives, which is right over here on the right-hand side. I'm gonna do a demo of that today. And these are our 3 highest priorities. So Hydrilla is right here, water chestnut, and Starry Stonewort. I'll definitely go more into ID characteristics. So this is a part where we figure out where you're sampling. If you're not sure yet, I put this chart together so you can make an informed decision. We do have a few people I know said in the multiple category, anywhere from hemlock to Canadice to Canisius. So those aren't zeros for real. But yeah, you can just figure out where you want to sample using that. We ask you to stick with the same sample spot. So what I'm thinking for this is that we'll just have you guys sample in the same exact spot every 2 weeks, and then I'm never opposed to extra data. So if you want to do other spots, you don't have to do that same one every single week. But if you're just like, oh, I wonder what's here, I have my rake in my car, let me do a rake toss, that's totally acceptable. The only thing that I would say is clean your rake, let it dry, maybe just give it a wipe down to make sure that nothing's hanging out. Sometimes you get some muddy samples off of rake tosses, and things can be spread that way. So that would be my only concern. I was hoping that this would be the first time that we've done this, but if you choose your spot, choose it in a spot that you're sure you can go to and every time, and send me the coordinates for that location. The reason that I ask that is simply because we have, we're going to be mapping throughout, like mapping our results at the end of the year. So it would be nice to have a set location, and then every time you go to that location, at least you can put in those coordinates that you had, so that they aren't, I'm sorry, I'm looking at the chat real quick. You can put in those coordinates so that I know that you're taking your one sample that you do every time, and then for the other ones, you can just set it from the map. And then you can sample there every week, or every 2 weeks, I'm sorry, within 20 feet or about 6 meters. So you could have a buffer around that point, but just enter that point as your location. So, and yes, Catherine, I was just about to go into how to get coordinates. So if you have an iPhone, it's really extra simple. You can just go to this compass app right here, and that will give you your coordinates. Those are going to be right there. That first line in the degrees, seconds, and minutes. Yeah. Also, if you don't have an iPhone, the other way I would do it is by Google Maps. So if you have a spot, let me see. I'm at the Finger Lakes Institute right now. If you just press a spot on the screen, it'll give you a little point, and then you can press on that point. Oops. And it pops up with information, and it's showing the latitude and longitude, not the degrees, seconds, and minutes, but it's the same difference. I can use either bit of information, and that will give you exactly where your spot is. And you'll only have to do that one time. Just make sure you're going back to that spot and save that number so that you're able to. Oh, true. It will also capture coordinates. Patty said the app last year automatically generated coordinates. Yeah, you can also do that. It will, yeah. If it shows, I'm trying to think if IMAP shows your coordinates automatically. I think it does. And you can definitely pick from that. What other? Oh, Lanternfly has been recorded in Ithaca. Dang. Mention the app Scott had this morning. Oh yes, thank you, Mary. I'll go into it more. Well, yeah, I don't have any slides on it, unfortunately, but there is one app called Seek. I'm putting it in the chat right now. We were doing that this morning, and Scott was using it for every single species that we saw just to check its accuracy, and it was really accurate. There was one fragment of buttercup with just a few leaves, and it was able to identify it and point us in the right direction. So if for identification, you can take note of that and look into that. It's basically you just point your camera right at the plant, and it gives you a name for it. Michelle, I don't know what you're saying. I'm sorry. So here's how to do a rake toss. I showed you the rake a minute ago. Here's another picture. That's how it should look when it comes out of the water. Well, not how it should look necessarily. That's how it might look. You might not have nearly as many plants as that. We did a couple of tosses, empty tosses this morning, that's for sure. Sorry. I would either one. I can do. I can enter either one of those, so whatever works for you. I think I would prefer decimal, but that's like by this much. It doesn't really matter to me. So can you guys see this YouTube video? No? Okay. I'm gonna make sure I'm sharing the right part. All right, now you can see it? Okay. Here we go.
Finger Lakes and New York State Boat Launch, and we're going to talk about how to do a rake toss. So first you're going to need 2 rakes. You can bind them together side by side with a couple of zip ties and a carabiner to the end. And you connect a rope to that carabiner. That rope is going to be 100 feet long, and it's going to be in increments of a foot between each. You're going to mark off each long section of that rope. So when you do throw the rake into the water, you will be able to judge just how deep it is. When you throw the rake into the water, you want to make sure you secure the end because you don't want to go in the water and not be able to retrieve the rake. So you pull it out and you look at all the plants that you have on the rake and you separate them into piles.
All right, let me try real quick and make sure that I'm sharing. Yeah, I don't think I'm gonna get any better with the sound, but here's the thing. Basically, she just said, I'll rewind it a little bit actually. 100-foot rope cut off or marked at each foot.
All of the plants that you have on the rake, you separate them into piles. Here I'm separating them into piles by species to see what I have present in the lake. And there's some Chinese mystery snails. This is some Algae. The same plant just looks very different. So plants can look very different depending on where they are in their growth cycle. And then we have this one here, and this one is the same plant as well, only it's...
Sorry, I'm working on it. Give me 1 second. I think that video really slowed down the functioning of my computer. Yeah, so I'll just give you guys like 5 minutes if you want to watch that real quick. And let me know if you have any questions. Yeah, the thing is, Carol, that it's just going so slow. Like it's oh, how did I do that? Oh, sorry, thanks, Caitlin. There we go. Sorry, I sent it directly to Caitlin by accident. John, you should have the link in the chat box. I'll send it directly to you too, just in case. Awesome, thanks, Caitlin. Brad, there should be a, yeah, I did stop screen sharing just so people could watch the YouTube video and to give my computer a second to recover. It was moving really slow. But yeah, the link is in the chat box, so you should be able to copy that. My computer was dying. That's why it was struggling. Glad I got there in time. All right, let me know if you're ready to keep going. Awesome, got quite a few people. Give it just a minute longer. Looks like I got burnt this morning. Sheila, I'm not really sure about cheap rakes. I haven't shopped for them in past years. All right, I'm gonna keep going. All right. So data collection, this year we're using IMAP invasives. Like I said, it's a statewide database, so everyone across New York State is encouraged to use IMAP invasives. It's a really good program. We've got every PRISM reports using IMAP. The mobile app is really simple, and then they also provide more advanced stuff for land managers, and you can view all of it just with a free account, which I will show you. If everyone wants to go to imapinvasives.org and make an account, that would be great, and I will show you how to, email me your ID number, please, and I'll show you how to, I'll have to show you how to find your ID number, obviously, so don't panic. Yeah, this will be the page that you open up to. Let me know when you've got your accounts all done, so you're not trying to do this and listen to me at the same time. Do that later. And they have different people running each state's IMAP invasives. Obviously, you'll be at the New York one. Ours is run by the Natural Heritage Program. Sweet, thanks, Sheila. And I'm writing yours down, Sheila, but if everyone could just email that to me. I've got a slide at the end of like follow-up stuff, so I just don't want to lose it in the chat if everyone starts typing them in and I close out of the Zoom. All right, so you'll find your ID number by clicking this little hamburger menu, the 3 lines up in the top. Go to your account. And this is your person ID number in the second box in the top left-hand corner. So then you'll be able to filter for, knowing that will allow you to filter for your findings if you want to see them on IMAP, and allow us to filter for your findings. I don't believe they send you an email with your ID number. You have to find it by going to your account and just checking this out. I'm actually gonna need to write mine down. All right, it's a different email, my IMAP Lisa that I gave you. No, it shouldn't mess anything up, Sheila. Because I'll be searching for the person ID number. All right, so I'm going to, oh, hey, someone already requested membership to the project. Nice job, Shelby. What are you waiting for an email for, Bradley? Sorry, it's been a while since I made my account, so I'm not sure if you have to confirm your email and such. I'm assuming so. Okay, gotcha. I'll give it a couple more minutes then. Yeah, check your spam folder, and then make sure you're refreshing your email. Don't see an account number, but I was able to sign in. Yeah, just go to this little menu right up in the corner and press my account, or your account. Let's see, I'll go back. Make sure we can see it. Yeah, your account. And it'll be the person number in the second box. I'm gonna sign in with my phone so we can see the mobile app. So there will be 2 Megans on Zoom. No, it doesn't have to be done now. I'm gonna pull this up on my phone and then move ahead, and we can always you can always message me later about questions. All right, and then you can download the mobile app whenever you get a chance. This will be, you don't have to use this to report necessarily. You can take photos on your phone and make notes of the information, which would just be like density on the rake, how long you spent surveying, your location, and then submit it online if you prefer that way. Yeah, so Lois, you're going to be seeing the map. I switched over to the mobile app right now, but I'm definitely going to go back to that. I just want to show you, I'm going to show you where you would enter like project information and other stuff. And I'm going to enter a data point, and then we're going to take a look at it. All right, so I have not been refreshing my data. I'll do that at some point, but it hasn't been a hindrance yet. So it'll pop up with tutorials. Real quick, you can just tap the screen and it'll go out of that. You can also go to preferences up in the left-hand corner. Here you'll be able to like change your jurisdiction if you ever left New York and wanted to report something. Default base map type, road versus satellite, metrics, default project. So you can set the project and the organization. Mine will be Finger Lakes Institute. You guys won't have an organization, but you'll have the project. Definitely. And I'll show you how to do that when we go back to the desktop version. The other thing that I wanted to show here was, customized species list. So if you go into IMAP, you don't have to do this. It's just a really long list of invasive species, like every insect, every mussel, every invertebrate, anything and every invasive species that someone might possibly want to report. So you can go in here and select what species you want on the list. And I will send out a list of recommended species if you want to do that. I'm going to make notes so I can do that later. But I've got brittle naiad, Brazilian elodia, mostly aquatic plants, hydrilla. You just press OK and then make sure to save down at the bottom. Oh, apparently I can stop them from showing me welcome instructions, which I'm going to do that because I don't need them. All right, so you've done your rake toss. You're ready to enter some data. So you just press add observation. So we're gonna do, you can only report one species at a time per observation. So the way that we've decided to do this is you can also report species detected and not species not detected. And it is really important to enter not detected data, especially for our top 3 priority species. So we're gonna ask that you submit 3 records for every Rake Toss for our top 3, Starry Stonewort, Water Chestnut, and Hydrilla. And then you can put presence or not detected, no matter what you put a record for it. And then any additional invasive species that you want would be like any additional invasive species you find, do an additional record for, and then we're gonna have you note, I'm gonna send out a list of native species and abbreviations for them, codes, like a 3-letter code, so that you can note in the comments what native species you found. So unfortunately with the mobile app, you're only allowed to take 1 picture, but we want as many pictures as you can possibly take. Like as many, if you think like, oh, this is a good picture of the leaves, let me take a good picture of the stem, let me take a good picture of the node where it branches out, let me take a good picture of the flower, any aspect that you think will be helpful in identifying plants, which we'll go over as well, we want a photo of it. And at least 1 photo is really important. If it's like a really clear photo, that works too, just 1, but it's really important because if you don't submit a photo with it, we have no way to go back. Every data point is entered as unconfirmed in IMAP invasives. So the only way for us to confirm it is if we see a photo of it. Otherwise, there's no way we can be sure enough to add it to the database as confirmed. It'll still be in the database as unconfirmed, but for people to be sure, we definitely need photos so that we can go through and confirm them. So you can pick a species. This is my list of species. It's not 100 species long anymore. And I'm gonna do a fake species. I'm gonna do a detected record, take a photo of something random. Oh, no, I'm not, because my phone's camera is not working. I'm not taking a photo of myself. All right, and then over here, so this, it does show you lat and lon on IMAP, which is even better. So you can just take a screenshot of that or write it down and then let me know what your point is and just return to that area within the buffer for every block of sampling. And then I would also ask that on your in your subsequent records, you enter that exact number so that it's the same and we know that it's within that buffer. Time searched, we're gonna want, so for the last record you submit, just let us know how long you spent on the survey so that we can get an idea of the volunteer hours that we're generating. It's a really important metric for the PRISM in general and like a measurement for success of our programs. Size of area containing invasive species. So this one is, I would just put the smallest one because it's just, we're just doing point sampling and whoever's looking at this, actually I don't think you have to put anything in there. So I already started to, so it's not gonna let me put nothing, but I don't think you have to enter anything there. So you can just leave it alone. Distribution of invasive species. So you can do this as a, it's not going to be distribution of species in that area, which is what this would be intended for terrestrial surveys, but it's going to be the amount of plants that you find on your rake. And I'm going to show that later as well. So then you add any comments. So you'll want to add native species. And you don't have to do this on every single one. If you're submitting 3 or 4 records, then you can just do it on one of them for that spot if it's the same rake toss. So like Eurasian water milfoil, or no, that's not native. Coontail, I think I made the abbreviation COT. Elodia, ELO. Whatever, you get the idea. I would also put any comments maybe on the amount, oh, the dominant species, that's what I wanted in there too. Dominant, submerged, dominant floating. There's not always floating plants. There's not even always submerged plants, but so you can feel free to leave it blank, but just put it there still so that we know that you were thinking about it and that there really wasn't a dominant, there were no floating species. Yeah, so that looks good to me. You can also add any notes about what stage it's at, if it's flowering. Some people look at the weather in these kind of surveys, so you could put that if you wanted to. Any other information. Oh, you could also put, like, sometimes when I would do these rake tosses as a steward, I would comment like there were clumps of zebra mussels on the plants. You can actually report zebra mussels using IMAP invasives as well. Yeah, so you'll just press save. It'll show up right here. You press the check mark. Up in the corner in that left-hand side, you can press upload selected. I'm not connected to Wi-Fi, but that's okay. And I'm sure I want to upload my records. So that's that. You can, if you take additional pictures too, I'm pretty sure I said this, but maybe not. You can add them, you can edit your observation online. So I'm going to show you how to do that as well. Let me stop sharing my screen from my phone and start sharing my screen from my computer. Anyone have any questions so far? Should I pause and temperature check? All right, looks like we're doing good. Okay, so now I'm going to go back to the map. Sometimes it takes longer to load than others. It's a lot of data, so these hexagons show that there is data there. There is some type of invasive species sometime in life, like anytime. Some of the records are really old. Someone has reported some type of invasive species. So if you scroll in, I might need to go a little farther than that. I'm actually going to filter so it doesn't take so long. So you can filter by species. You can filter by project name if you want to see everything that people are submitting to our Macrophyte survey or have submitted. I'm still got to show you how to become a part of a project. Habitat type, genus, any of these things if you filter. So I do water chestnut pools, so I'll refer to this for information on water chestnut populations. And if I apply the filter, and scooch down in here, those surely are not all water chestnut. Okay. So yeah, you can use this. It'll also, you can go over to layers on and off. And if you want to see treatment records, we've got some treatment information that you can't really see, but there's a little gray speck under there's a population or treatment that people have done hand pulls for water chestnut. Oh, we got a couple notifications in the chat. Will you review our first submissions and let us know? Yes, definitely. It might take me a minute, but I will do everything I can to get back to you within a week. It's definitely, I totally understand the importance of that when you're starting out this type of program. And you can also see not detected data for water chestnut. So this is all water chestnut. I'm not going to go through this because, well, it's quick. So you can also, anything that your filter, if you go to export, if you want to you want just a report in a chart, the Excel file, CSV is basically the same as an Excel file as far as I know. And you can, this will export based on your filters in the extent that your map is showing. And it'll just give you a whole chart of what's going on. All right, so find record allows you to, let me clear this filter real quick. This allows me to put in, oh, it goes by record ID. I definitely thought it went by person ID. So your record will have an ID, but I'm just going to do it through the observer. And we'll see if we can see my fake species submission. We can't because it's not confirmed. So it's only going to show you confirmed species unless you specifically toggle this on. And there it is. If you press on it, it will pop up. And it was submitted today, June 5th. You can press details. And then this is where you can, so I have the ability to confirm aquatic listings, but you won't. And then, what was I about to do here? So this is where you can add in pictures, I believe. Yep. So add photo. There weren't nearly enough pictures with that one. I didn't get those leaf margins. Couldn't get a good picture of the root. Let's see. Oops. Sure, there's some downloaded pictures. And then you can just, if you have them on your phone, you can just email them. There's Dave, our Starry Stonewort guy. You can just email them to yourself and then save them. And then post them. And you're able to edit. On the online version, there's a lot more detail too. So if you have any other details that you want to specify for us, that is also beneficial. And then I'll just press save. And now my record's got another picture. All right, let's go back to the map. Let's check chat. My email was also in spam. Yeah, so definitely check your spam. The site doesn't like can't log in. All right. Sounds good, Jill. I'm sorry about that. Sometimes IMAP is really slow, so it might be, it might just be them. Can we go in and do our own fake species? Definitely, definitely, definitely. So I'm going to show you how to do the projects. So same hamburger menu up here, you just press projects. And our project is Finger Lakes Macrophyte Survey. And you'll be able to, up in this corner, I believe, there's nothing there for mine, but there will be a request to join box. And then I will go ahead and approve the ones who requested already. I just got another one too. Let's see if it refreshes. All right, gonna refresh one more time. And then I'll move on. Oh, you guys are getting it. My email's going off. All right, and I'll keep this window open if anyone else is dying to be added. I can come back to this. Okay, sounds good, Mary Rose. All right. So I'm just going to show you quickly how you can enter your data online if you prefer to do it that way. So you can just press create record. Presence data. Actually, yeah, so presents or not detected for the if it's top 3 and you didn't get any of them. But I'm gonna do presents. Same thing, you can enter coordinates just like before. So if you're doing your primary location, then enter those coordinates. If not, you can just put a point wherever any other new spot. You can draw a buffer if you want to, if you went up and down that area. And it's basically all the same stuff. You enter the species, enter the project. I gotta pick a species. It'll ask you for photos. You can add as many as you want. Put the same comments for native species, time spent looking, what's in advanced details. And then you can also go through here and see if you want to add phenology. Was it flowering, budding, just a rosette, other information. And it's the same process. You just pick complete. It'll be entered as a non-confirmed data point. I'm not going to do that. All right, anything else I should look at on IMAP? I believe that covers it. Projects, species list. All right. Great. All right, so this is just confirming all the stuff I pretty much already said. For each sample, submit at least 3 records, 1 for each of the following species, detected or not. Submit an additional record for each additional invasive species found. So if you find some Eurasian water milfoil, some curly-leaf pondweed, European frogbit, brittle naiad, any invasive species, let us know about it. We still want to know. It's just not as important to do not detected, then you'd be entering records for a long time. In the comments, note native species. This is exactly what I was talking about before. I'm going to send you a list of shortcuts, which I have not made yet, so that's why you don't have it. Additionally, in the comments, make note of water depth. Water depth, definitely. Water depth is useful. And any other relevant information, like I said. Photos, submit more online if you need them, as many photos as you can or want to, however many you think you need, for us to be able to do an accurate identification. And in your final record submission, please note the time you spent under time searched, time you spent surveying. Are we able to use the map to see what areas are covered by volunteers? So if you filter for our project, it'll pop up with what's been submitted by Macrophyte Survey. You would probably want to filter by year, so 2021, from May of 2021 on, and that'll give you what's happening now. All right, so this is what I meant about the distribution or density. So this would be a trace amount if you come up with your rake with just a couple. Sparse is a little more than that. I wouldn't say that's medium. I would say that's dense. These are a little off. I've got better pictures right here. Oh, no, I don't. So this is the idea. I do believe I had better pictures though. I'm gonna actually go back. So generally it's, non-trace, sparse, medium, dense. So trace is just like a plant or two. Sparse is, so they say trace is 1 handful, sparse is 2 handfuls, medium's an armful, and dense is just a covered rake. So I would say that this is sparse amounts of plant. I'd say this is medium, and I'd say that's dense. That's what I'm going off of, at least. That other slide was not what I consider those to be. So this is going to be our schedule for the season. If you have a rake already, start whenever you want to. We're going to start the next block on the 14th or on the 21st. Hopefully everyone will have their rakes by then. If it's looking like we won't have our rakes by then, I will see if I can do an office day or do some driving to get people their rakes. If you guys want to meet me at the FLI, or if we can figure out a meeting point, halfway spot, we'll figure something out. That would definitely be ideal. No guarantees. It might just all go in the mail. That would stink. But it's a possibility, unfortunately. So I'm going to be sending out a newsletter the week after the start of the block period. It's going to contain information on what species might be popping up that weren't there before, interesting finds, what people are finding and some volunteer photos. Also events that the PRISM's putting out, I'll probably stick in there. We've got a lot of water chestnut pulls throughout the region. So if you're interested in coming out and doing that, it's usually a really fun time. Super satisfying to see all that water chestnut disappear. I'm gonna do my best to hold office days on Tuesdays. My schedule right now, we have a water chestnut team who's also doing watercraft steward stuff. So they're gonna be working Wednesday to Saturday. And I'm planning on working. There are some Saturday events that I'll be at. So if there's not a Saturday event, I'm planning on being in the office on Tuesday. So I'll let you know. There will be at least one office day per block, but I'll let you know what that's gonna be. I think there's only 1 or 2, maybe 3 Saturdays that I'll have to be working. Honio Lake is shallow where I live. Can't I just walk out in the water with my garden rake? Yeah, yep, definitely. Our water's shallow. Should we pull horizontally? Yeah, if you want to go in, if you want to just grab the water, if you have waders, you can walk in, or if you're just down to walk into the water, I'm definitely cool with that. You can still report what you've got. And if you're doing that, then I would say include the just do the distribution as kind of what you're seeing in the water. Our office location is in Geneva, the Finger Lakes Institute out of Hobart William Smith, 601 Main Street to be specific. Do you have the rakes available now if we want to go to the FLI and get one? The thing is that we only have a certain number of rakes and we kind of need them for our certain programs that we have planned and for our field team, because Hydrilla has an outrageous number of people. Our Hydrilla team, it's not outrageous, it's good that we have so many people, but the program kind of got merged with another program, so they've got some interns and some employees. The interns are from the DEC, I believe, so we just, like, their main thing is doing rake tosses. They go out in kayaks all day and do rake tosses and look for Hydrilla. So they definitely need them, and we need them too for water chestnut, because we take rake tosses whenever we go to a certain site. Any other things I missed? Oh, but also by office hours, I don't mean you have to come see me, Mary Rose. I just mean, like, I'll be available to answer questions pretty quickly. Are we to create our own rake, or will we? The rakes will be provided. So we had a contract with Idea Collaborative. These people are going to make rakes for us, but being funded by New York State, there are certain, like, guidelines and suppliers that we have to go by, and we just reached some roadblocks that we weren't expecting. So that's what's causing the delay. As soon as we get them, they'll be out to you guys. It's definitely my highest priority. But if you guys do want to, you can definitely do surveys by just getting in the water. Is there a guide for identifying what we find? Yes, there is. I'm gonna send that email out to everyone. We've got a really good field guide. If you're coming to Dean's Cove today, I'll be able to give you one. But otherwise, I'll send it out. We have a link online. It's all online too, created by the PRISM. It's got native species, invasive species, invertebrates, terrestrial plants. It's a pretty comprehensive guide. So I'll send that to you guys as soon as the training's over. Just making a note of that. And yes, I'll do my best to respond within 3 days. We also have another coordinator who's a good portion of his job is going to be dedicated to checking IDs, IDing submissions and stuff like that. So we should be able to do better than we have in past years. What's the latest publication year for our field guide? I am not sure. I think that it's 2017 at the earliest, but I don't think it's that old. Definitely got all the species that you would expect to see. Can you please share a copy of your slides from today's session? Yeah, yep, I'll send out the PowerPoint with the recording. Can you repeat the name of the ID app that used the photo? Seek, S-E-E-K. All right, so we've got a Facebook group. If you guys have Facebook, check it out. I can just request to join and then I'll approve you. This is so that if I'm not getting back to you fast enough, you could post, or if I would email me and post it on Facebook, so you're not waiting for me and then waiting for people to respond. But this is our Facebook group. You can post any findings you have questions on. Also, I'm asking that you put hashtag your county name, MSP, on there so that it's there. That's also in a post in the Macrophyte Survey Program right now. So it'll give examples, but this like Kyuga, MSP, and that will put your discussion into topics so that you can go and check out specific counties if you want to see what people are finding, where people are posting about near you. And there's probably going to be like Hillary, my boss's boss, has the director of the PRISM. She has access to this page. She's the admin too. So she'll probably post some stuff here and there if she gets a chance. I'll try to post some stuff just on happenings of the PRISM. Save the date. So this is like I said, the Starry Stonework Collaborative happens to, yeah. I'm sorry, Bill. That totally slipped my mind. Yeah, definitely. Any and all ID resources are helpful, especially when you're first getting started. I was telling the people this morning that even like I have experience IDing plants, and even when I first started as a steward, there were some of the most obvious plants looking back now that I was just like, not so sure. It still looks a little different than this guide. So yeah, definitely. I totally get that. Use as many ID guides as you want, and let me know if you are not sure. Also, like I said, in IMAP, you can confirm or not confirm species. So you can submit species that you're not sure about. Like someone will go through and confirm it or be like, no, that's wrong, and correct it. All right, so this is part of the Starry Stonewort Collaborative. So the River Alliance of Wisconsin started doing this, and then Minnesota Aquatic Invasive Species started a few years after that. Starry Stonewort's a big focus in the Great Lakes Basin, and they have a couple events throughout that day to demonstrate to people how to sample and then send out people to sample on that same day. So we're trying to do a version of that, basically the same thing. We're going to have an extra steward at a couple of the busier launches, hopefully, so that people can get their demonstration. And I'm sorry, I got distracted by the chat. So we're going to have people at busy launches doing the Finger Lakes. The Finger Lakes watercraft stewards are going to be doing demonstrations so that we can have people. So if you want to go take as many samples as you can, get as many water bodies as you want, bring people along, let them know what's going on, what you're looking for. Especially you guys would be a huge help in this, because if you do bring people, then you'll have experience by this time. You'll be way more confident. You'll be able to show them what's up, and hopefully we'll get more people involved. All right, I understand that we should all use the same project title, but do we need to use our own primary organization name? No, you shouldn't have an organization name. You have to be approved by, and I'm sure Hillary probably has ability to approve, but they also, like the IMAP people themselves, will approve PRISM names. They usually, like when I first got my official email address and signed on, they met with me and like added me to my organization. So as volunteers, you don't need an organization name, but we definitely want the project name. All right, so this is our last section. Some ID. So a macrophyte, which I probably should have had earlier in the presentation, is an underwater plant that you can see. Here are some other references if you wanted to get your own field guide. Like I said, main field guide of invasive aquatic plants is probably very similar. I didn't make this slide, someone else did, but I wouldn't be off put by that. Saying that it's from Maine. But yeah, like I said, I'm gonna send out this PowerPoint so you can always refer back to it. So when you're looking at plants, one of the first distinctions that you're gonna make is is it emergent, is it floating, is it submerged? So water chestnut is a floating plant. It's got these little bulbs on its the petiole of the leaf. That might not be the right word. Don't quote me on that. That has a air socket. So floating, these lily pads, anything you see floating on top of the water is a floating plant. Pretty self-explanatory. So Hydrilla is a submerged species. Starry Stonewort's a submerged species. Elodea is a submerged species. Mostly what you're going to be getting is submerged species. If you see floating species, you don't have to get it on your rake to record it. So you can just add that to your native plants. And maybe take a picture of it. If it's a lot of floating species are native, but if it's an invasive, then definitely take photos of it. So you want to look at a variety of aspects of a plant to determine its identification. Oops, sorry. Leaves can be arranged in a different way on the stem. It's in that next slide. It's going to be a better way to explain that. How many leaves are there? How many leaflets are there? Margins are the outside of the leaf. Is it toothed? Like water chestnut has tooth margins, but a lot of pondweeds have really smooth margins. Stem shape, we've got a flat stem pondweed that can help you. The Starry Stonewort is a macroalgae, so it doesn't really take root. It has hold fast, but it gets its nutrients from the water column. So that has a really round stem and you can pop it if you squeeze it together. Roots, are they strong? Are they weak? The roots of a water chestnut, I went to the Erie Seneca River by Montezuma yesterday, and the water chestnuts are just starting to pop up, and you could easily pull them right out of the water. You could see the little chestnut at the end. Flower color, that's the best indication for any plant is to be able to see its flower, but unfortunately that's not the case most of the time. But you can look at color, number of petals, and then the seeds and fruits are just as important, I would say, as the flowers if you have them. They always say that they want you to have like a full sample from roots to the apex of the plant, flowers, fruits. It rarely ever works out that way. These are some leaf shapes. This is what I was talking about, the arrangement of the leaves. So opposite or alternate, they stagger along the stem. Opposite is right across from each other, and that's only 2. And then if they're coming out of the same spot and there's more than 2, that's considered whorled. So Hydrilla has whorled leaves of 5. We saw a buttercup today, which is native. It had alternating leaves. And then what has opposite leaves? Can't think of anything off the top of my head, but you can definitely, it's recognizable. And here are some also leaf shapes, which I admittedly am not the best at. And this is showing you the reason there are two aspects of leaves shown for the arrangements. So when you're looking at the leaf, it's a one leaf and made of leaflets. So this has leaflets on it. The column on the right, these are little leaflets, pinnate, I would say. But it's still considered one leaf. So the buttercup we saw today had 3 leaflets. So I was really confused at first because it said alternate in our guidebook, but I was like, this looks like it's whorled. They're all in the same spot. But then we got a better sample that you could see the branch and then the 3 leaflets popping off of it. So only one thing was connected to the stem. Stems, like I said, you can look at so this is actually, I can't remember what the name of this pondweed is, but the stem actually zigzags. There's a better photo in there. Oh, actually, I think this is white stem pondweed. So the stem's white. And there's a flat stem pondweed, stem's flat. Here's some roots. So these are floating roots. I can't remember what they're called, but the same, if you've seen mangroves in Florida, they put out their roots that just take nutrients from the water column. Same difference. They got some rhizomes here. They'll take root, send out a shoot, take root, send out a shoot. That's eelgrass right there. And here are some flowers. I believe that's Elodea. I've actually never seen Elodea flowers, so I'm not 100% sure. I've definitely, yeah, I think that's what it is. And then this is Sago pondweed. So flowers can be arranged in different ways too. So this is a solitary flower right at the end of the eelgrass. And then this is from Sago pondweed is a raceme, I believe would be the correct term, where everything's on kind of one stem, multiple flowers. This actually looks like, I'm not sure if these are flowers or fruits, but the fruits replace the flowers. So that would be the arrangement. All right, and yes, some distinctive fruits. The Starry Stonewort has these cute little stars on them. If you're lucky enough to get a sample with that, that's an extremely unique feature. So you know for sure that's Starry Stonewort. This is water chestnut, already a really solid, easy to identify plant, water chestnut in general, but if you see the chestnuts themselves, there's no question about it. And then this is horned pondweed, and it's got little horns. So if you see those, you know for sure that's what that is. Otherwise, it might be a little difficult. And these are turions. They're also, curly leaf pondweed has these as well, and it's just so that's what they sprout out of and put roots down, fruit. All right, and here is our main enemy, Hydrilla. So the Hydrilla generally in our region, Hydrilla has 5 leaves, so whorls of 5. And the main defining factor is those little tooths on the edge of the leaf. So they are super fine. Take a look at them if you think you found it. And we're also going to provide small microscopes in our kits, so you can use those to check for those really, really small leaflets. These are the tubers. Tubers hang out in the soil for a long time. We've done some they do some tuber sampling to figure out and try to sift them out of the sand in some areas. What else? Yep, tubers and turions. Tubers are small potato-like structures. Small potato-like structure. So this is just another look at, oh, well, it's asking a question, but I'm going to tell you the answer. I just told you all the answers, so. Oh, I guess that's the only question. Starry Stonewort. This one's a little harder to identify. The first thing I do is pop the stem. I know that it's kind of like, oh, am I going to realize the pop when I first see it compared to other plants? You will, like, definitely. I wasn't sure either, and the first time I saw it, I popped it, and it basically, like, it looks like they're just like repelled from each other. You just give it a little squeeze and it just pops right apart, way more than any macrophyte. So this is a macroalgae. Give me 1 second. All right, Starry Stonework grows up to 9 meters. It's got rhizoids in the sediment, and it puts out those little stars, like I pointed out. Whorls of 4 to 6 blunt branchlets. So these are the whorls. This is a whorl. This is a whorl. This is a whorl. They put out, so they'll branch, and then they'll branch some more, and then they'll branch some more. That's how it gets so thick and dense in the water column. And it is hard, it's a little difficult to identify. It's got clear rhizoids in the, so if you're able to get the root, it's got clear kind of filamentous roots. And also they call them like crazy arms. The Starry Stonewort, the length of the arms are never really exactly the same. So there's the stars, and there's, you can see just how dense it really gets. Actually, does that have? Yeah, no good pictures of the roots, unfortunately. Oh, describe where to squeeze. So any part of the plant, any stem. The stems are hollow, so they, because they just take in nutrients from the water itself. Macroalgae do. So any part of the stem you can squeeze. This is water chestnut, super easy to identify, floats on top of the water. You can see these little, these are the rosettes that are coming off of this one. Triangular toothed leaves. Water chestnuts themselves are really spiky. They're sharp too, so be careful. The leaves themselves, they've got different types of submerged leaves, and they kind of just look like, you can't really see them here, but they're like thin thread-like leaves, and they just kind of sprout in every direction. They sprout out of the stem, and then they sprout like, oh, just like the world where I was showing you the leaflets. That's basically what they look like coming off of water chestnut. Cord-like stems, roots are feathery. The root is gonna have the, if you get the whole plant too, you should be able to see the water chestnut that it came out of. And these guys survive, the seeds can survive in the seed bank for like 15 years. So that's just a fact that I didn't note. And the petioles, that's let's see, we got a good picture here. Sorry, I'm constantly moving around the Zoom windows. Wow, there's really not a good, oh, you can kind of see here, it's a little inflated on this leaf right under that bug. He's in the worst possible spot. But usually it'll be right here, like along the stem of the leaves. Curly leaf pondweed. This one is a invasive species. It's pretty naturalized at this point. It's not a high priority, but we still want to know about it if you see it. This one does get a little more interaction with native species, so I would say it's not as bad, but it does grow early in the season and can shade out a lot of native species. When I say interaction with native species, I mean like invertebrates and fish, not other plants. Other plants it definitely will shade out. Alternate arrangement of leaves. They call the leaves lasagna leaves because they look like little lasagna pastas. And yep, alternate. They've usually got a clump of leaves up top. And these are the flowers you can see also in that raceme. Small reddish green flowers. And anything else I'm missing? Are the water chestnuts the same kind that are panared? No, they're not. Unfortunately not. They are used in their native range as food sometimes. You can grind them up and use them as flour, dried out ones, but it's like not allowed in the US because they're known to collect heavy metals in the water column. So just for safety reasons, they just say no in general. Yes, please do not plant them. That would definitely be not great for the program. Eurasian water milfoil is pretty much as common as curly leaf pondweed. This guy does get really, really thick and it's everywhere. So there's a native water milfoil called northern water milfoil, and it looks like this, but you can kind of see how this is kind of a flat end. These last few leaflets have a flat, blunt tip, and the native would have a tapered tip, a little more clear than this. The leaves for the invasive are also larger. It's just kind of a bulkier plant in general, and this one doesn't. The native, sometimes the leaves will stand up more, like they hold more rigid outside of the water, and Eurasian water milfoil just collapses completely onto itself. It's got 4 whorls, leaves of 4, and usually you can tell also because some of them will have a really reddish stem. It'll be on the stem, and then if it's still growing, this top apical branch will have a red base too. All right, European frogbit. It is not super common in our region, so definitely want to know about it if you come across it. It's a floating plant. Those little leaves are about this big, I would say, so you're not looking for any large lily pads. They are free-floating. They have kidney or heart-shaped leaves. They're really rounded right here, also at the base of the leaf. The stem lacks a midline, and it's got 3 petals on its flowers. All this little stuff around the European frogbit is duckweed, which is native. It is not the frogbit itself. Brittle naiad. It's got some spiky leaves. It's usually curled. Looks like it keeps it. I've actually never seen this in person, but it's supposed to keep its rigidity pretty well. Have to hop off. I'll watch the rest via recording. Thank you, Caitlin. Have a great day. So brittle naiad, it's got those curved leaves. They're opposite, and yeah, I think it does say it's stiff specifically. Seeds are purple and curved, and they look similar to native naiads, but it's got those. This would be another one you probably want to use the microscope for, because the serrations are pretty inconspicuous. All right, now we got some natives. Coontail is a really popular one. It holds its shape outside of the water, and it's got these bushy ends to them. It's really, this was the one species that I was able to 100% confidence report by myself, because it does really hold its shape outside of the water. And it's got these really distinct leaves. So if you separate out just one of those leaves, it splits in two. You can kind of see right here, that's one leaf, and they're all in whorls. What time is it? Oh man. All right. No worries. We're getting through it. We're almost done. I swear. Cara, this is another macroalgae, so it pops just like the Starry Stonewort. That's what makes Starry Stonewort hard to identify, because it's got a lot of lookalikes. Cara, there are different types of Cara. Muskgrass itself has a smell to it, so if you're not sure and you smell a skunk-like odor, you'll be able to say for sure that's muskgrass. But there are other Caras that aren't Starry Stonewort, and they have the branches are just a little more even. That's the main difference. And obviously there's no star bulbils. Eelgrass is a popular one, super easy to identify, really, really straight leaves, smooth margins, parallel veins, and then when they're flowering, they've got these really, really cute little curly cues. So that's what the flowers pop up at the end of. You'll see a lot of that, like it says in July. Native pondweed. So curly leaf pondweed is the only pondweed that is invasive. So if you see anything else that looks kind of like it, and you're not recognizing it as a invasive species, then it's probably a pondweed. If you think it's an invasive species, feel free to report it as whatever you think it is, and we'll confirm it if it's not. We'll confirm it or not confirm it. So white stem is the one we saw before. It's got those zigzag. Sago pondweed's also pretty popular. The leaves are really thin and thread-like, and it branches off from that one leaf. It's got a bunch of leaflets at that node. All right. So be on the lookout for the list of abbreviations of native species. Send me your coordinates and your IMAP ID number. I did write down 3 ID numbers, but any of the other ones I didn't write down. Please just email them to me so that I can make note of it. If you haven't joined the Facebook page, go ahead and do that. And also be sure to join the project and start whenever you can. We'll get the supplies out to you as soon as possible. I have to go to the bathroom. What about algae? We get a lot. I don't know specifically types of algae. I'll look into like what was Kate, our Hydrilla program manager, knows a couple different types of algae. I know there's like one that's like really clumpy and it's just like gross, pull apart. I think that's called water meal, but I'll send out an email and see if I can get you any information on types of algae that you might see or might come into your lake. No, you don't report them. Join the HABS program for algal info. Yeah. I'm not sure what exact HABS program you're thinking of, Mary, so maybe you'll want to specify for people. Give me 1 second. I'm going to go to the bathroom and I will be back for questions. If you are, you don't have any questions, then you're good to go. I'm going to send out this recording and the slides to everyone and some of this other information. And I will see those of you who are coming to Dean's Cove around 1 o'clock, maybe a little later. I'm definitely going to take a half hour to eat some food, but it's 20 minutes from here to the FLI, so I should be able to be there by 1. Thanks, Janet. Have a great one. I'll be right back to answer any more questions. All right, I'm ready for questions if anyone's got them.
Can we ask orally?
What was that?
Can we ask questions orally?
Yeah, definitely.
Okay. So if we're not on Facebook and never will be, does that inhibit our participation?
No, if there's, you just won't have access to the groups. I was thinking about starting a Slack page, which is another program where you can communicate with people. It's the same exact thing basically as Facebook group, but I just figured if it wouldn't make sense for us to have two platforms because the second one, like people are just posting half here and half there, then it's not getting to everyone. Do you have a friend, or does your wife or someone have a Facebook that they would be willing to join the group or something like that?
No, and I don't want to ever go near Facebook.
Okay, heard. Yeah, so unfortunately, I don't think that you'll be able to have access to that stuff, but I can definitely, you can still definitely message me with questions, and I'll get back to you as soon as I can.
Okay.
Yeah, I'm
well,
sorry
I mean, I would use email for questions, but as far as collecting and reporting, all we need are the IMAP Invasives app, right?
Definitely. Yep. Mm-hmm.
Okay. So it's just I can't see what other people are posting and so forth if I don't use Facebook.
Yeah, but I'm still gonna send, I'll probably post the newsletters on Facebook, but I'm also gonna send them out by email. So you'll get like any interesting finds, like, or what people are doing, what kind of stuff to see, that to be looking out for in this time of year. So you're good. You just won't be able to see like the community-based stuff.
Okay. So what should we expect in the way of a packet? We're going to get links to the identification booklet,
Yes.
and a link to the recording of this session, and a copy of the slides from this session.
Yes, all of that.
And
the kit is going to contain, it might contain a couple other ID books. I'm not sure if we have those, but it'll definitely contain a physical copy of the field guide that I showed. It'll have a binder for like just about the program, like it'll have, which has information that you can refer to for IMAP invasives, so stuff like that. And then also it'll send, like, I'll put in that paper with the abbreviations for native species and just overall information about the program. So
okay.
that's kind of stuff
and
that'll
it'll come in US mail?
Yes.
Okay. All right. But all right. I think I've already told you I already have a rake. Yeah, so I don't need a rake. This is John Abel speaking. Okay.
Okay, perfect. Yep, I'll double check. I'm pretty sure I've got you down though. If you've emailed me, it should be in there.
Okay, but the packets haven't been mailed yet, right?
No, unfortunately not.
Okay.
I'll definitely let everyone know when they're mailed.
Okay, and so theoretically we're starting anytime, but probably the second block, which starts June 21st, is a logical time for us to start, right?
Yes,
okay.
Exactly.
Okay.
So when it comes to figuring out where we're going to sample from, if we want to go someplace where other people aren't,
Mm-hmm.
how do we know like I had said that I wanted to do the Owasco Lake Inlet, but I see 4 people are signed up for Owasco Lake, and I could be someplace where they're not.
So I didn't actually include inlets or outlets as the part of the lake. So so that would be perfect. Doing the inlet, that would be awesome.
Oh, okay, okay. What do you mean you didn't include it?
Like, so there's a other column too, like other that aren't the main Finger Lakes, and then undetermined, like people who gave me multiple options. So so the Owasco outlet would be in the other column.
Okay. The outlet.
All right. So Dean's Cove. I'm sending the address for Dean's Cove right now. I know some of you want to get on the road ASAP. US, New York. 14541.
We have some questions in the chat.
Yeah, yeah. I thought the Dean's Cove one was, I needed to get that for. All right, Bill Foster. One thing I'd like to bring to the table for consideration. I think we need to be careful of messaging regarding interstation. They are typically having no nutrient value. That's not really accurate. We need to start articulating on more system-oriented perspective on this stuff. Each of the species, once it is introduced, transition process. Ultimately, the system around it will balance out. Pretty much every species belongs in invasive species. Yeah, that's a good point. Just like that's what like I was saying with the curly leaf pondweed, that it's an invasive species, but it's kind of naturalized. So when I say naturalized, that's like no one's really going after it. It's everything's kind of adjusted. It doesn't really. It does crowd out native plant species, but other species are willing to like eat off of it. So yeah, definitely there's a scale. That's why we have our top 3 highest priorities, so that they have huge impacts and they aren't so widespread that we can't control them. So that's why we want data on detected and not detected. Catherine, yes, yep. After Coon's tail is native. Everything else was native, stuff that you would be seeing. Online ID source or ID plants before we get in the kit. Yeah, I'm gonna send you the field guide.
You said after which one was native?
After Coontail. Well, Coontail was native. Anything after that was native as well.
Okay. That's good, because some of them I recognize having seen for years.
Yeah.
I'm glad those are native.
Yeah, not all bad. Yeah, like I showed in that map, there's a lot of natives out there too, so.
And then how far do you drag the rake along the bottom? It seems like that would impact what you say for density, depending on how far you drag it.
Well, that's why we're just basing, because that's going to vary a lot. It's going to be hard to like determine that, especially if you're doing it from the shore. So like I'll just throw the rake as far as I can and pull it in after it hits the bottom. So I'm not really measuring that distance.
Okay.
So you just report what comes in on the rake itself.
Okay.
All right. Online ID source. Yep, gonna send that field guide. Sent out the address. Identifying species is part which I'm least confident. We'll be notified if we misidentify something so we can learn for the next time. I'm gonna do everything in my power to make sure that that happens. I realize that plant ID doesn't come naturally for terrestrial species even, and aquatic is that much more complicated because they're not coming out in their form where they in the water. A lot of them collapse once they get out of the water. I didn't address that we're gonna also send in the kit some little bins so that you can put basically like a little Tupperware. You can put water in and then look at the plants in the water, so that'll help spread out the plant material. But yeah, I'm gonna do everything I can to make sure that I'm giving accurate feedback. I know that we've had struggled with that in the past once the season really gets ramped up. But yeah, it's a high priority on my list. How far do we drag the rake across the bottom? I already answered that. Probably be there at 1:32. Sweet. See you then, Sheila. And thanks, Mary. Have a great day. All right, is there anything else?
Yeah, I don't understand how comprehensive our reports are. You say we should submit 3 reports relating to presence or non-presence of the 3 major invasives. And then on one of them, we should list what other species we found in our Rake Toss.
Yeah. I'm sorry.
Okay. And so we just need to make our best efforts to identify these additional plants.
Exactly.
But, you know, I think someone else has said they feel less confident about identifying. But do you want photos of all of the things we've included, or mainly of the invasives?
That's a really good question. Mainly of the invasives. If you want to know what it is and it's a native species, you can just send me. I wouldn't include it on the report because it'll probably be confusing, like if anyone else goes to look in years to come. But you feel free to send me photos of native species. For those that have Facebook, you can put it on Facebook. I'll respond to those. So yeah, definitely let me know. I want you to be confident in your reporting. So the reason for that we're really trying to transfer to IMAP invasives, like we're being pushed a lot to do all our data in IMAP invasives. So software is not specifically designed for the way that we've recorded data in the past. In the past, we've had all the native species and invasive species as option, and you just check them as you go on one survey. But I'm really confident in the way that this is going to work. It's just that native information, which for our data isn't as important as the invasive species, but if definitely I understand you wanting to know what it is. So yeah, just reach out for the natives.
So Bob Johnson does systematic surveys at the southern end of Kyuga Lake. I think he's on contract with this Tompkins Soil and Water and part of the Hydrilla effort down here. Does that data get sent to you, or is that?
If he's reporting it on IMAP, then we'll be able to see it, but I don't think he's probably in contact with Kate Monticelli, who's our program manager for Hydrilla. But I don't get it specifically. I'm sure they're probably in contact about it.
Oh, okay.
All right, I'll be on here a couple more minutes. Probably close down at 12:20 if there's no more questions or comments.
Megan, do you have anybody doing Myers Park on Kyuga Lake?
Let me check.
There's a boat launch there, so it seems like some trouble might start there sooner than just random lake spots.
Yeah, definitely. Let me check right now.
Well, in Finger Lakes Marine at Myers Point, they've had a lot of hydrilla within their basin, and that's being treated, I think now regularly. But I think that's separate from the park itself. So the Kyuga Hydrilla project is, obviously looking very closely at that side of Myers Point, the south side where that marina is.
Looks like I don't have anyone that specified it. We do have like 24 people on Kyuga that just said Kyuga. So maybe one of them is. But we definitely got the most sampling on Kyuga, which is really exciting because that's our highest water body of concern because it's got hydrilla in it. But yeah, Myers, I'm not specifically sure about.
Okay. If it turns out, I need to try it and see how it works, but if it turns out I can do it, is it possible to send in reports from 2 locations, or does that confuse the system?
Yeah, if you're gonna just make sure you do the same process, like top 3 species and then any additional invasive species. If you want to do other points consistently and you're confident that you're gonna want to do them every single 2 weeks, then you can send that location to me as well. If you just want to sample random spots, you can just send that in with the location that generates on IMAP. It's up to you.
Okay. Yeah, I'll do this Lansing Station Road that I signed up for, and then see whether I can do Myers or not once I see how much work is involved.
Sounds good.
Thank you.
Thank you.
So I signed up for a sampling on Kyuga Lake, but I don't have a location yet. But I live down in the Ithaca area, so I was thinking someplace close to Ithaca. So can you give feedback on what areas are needed for sampling, so to help me choose a spot?
Well, we've got a lot of people in Tompkins County that said Cayuga Lake, so I'm assuming it would be closest to Ithaca. I don't think that I haven't gotten direct like points where people are gonna be sampling. I'm pretty sure there's gonna be quite a few over in Ithaca. Maybe if you wanted to do like there's Ithaca, there's like a Cayuga inlet over there, right? Maybe if you wanted to sample there.
Somewhere along the inlet, then pick a point there. That would be, or the other place I was thinking is Fall Creek, which is quite wide there and is almost like the inlet, where which is by Stewart Park, basically.
Sure, yeah, that works.
There's some pedestrian bridges there. That would be a place that you could throw from. That's a possibility.
Yeah.
So when you get the coordinates though, if 2 people have like close to the same coordinates, then should we be picking another spot, I guess is what I'm thinking. So the points are not so close together, or, maybe, I guess I'm thinking, where is the need? Because I'd like to do a needy location.
Yeah, I'll try to send out, once I get those coordinates, it should be pretty easy to just throw it onto a map and see, so.
Okay, yeah, that's great. I'll get back to you about that too. Because I can tentatively pick a location, but then change it if someone else is sampling too close to there.
Cool,
If that could work.
Yeah, Louise, this is John Abel. I'm with the Kyuga Lake Watershed Network. And,
okay.
I know that Fall Creek is being watched for the Hydrilla project.
Okay.
And, you might want to try to tune into, what the Hydrilla volunteers are doing down there. And, I think if you sent an inquiry into Hillary Lambert, who's the watershed steward, she could probably advise you about that.
Okay, yeah, actually I do get some of her emails about various things, and I'm sure there's been one recently about the Hydrilla project, but I think that's a good idea. If I query her, then she can suggest a location that nobody is being infrequently sampled or something like that.
Right. I know there are a lot of people professionally and otherwise surveying the southern end and the inlet, and also Fall Creek, because we've previously found hydrilla in those locations, and they're looking to suppress it. The other person who might be able to advise you about good locations is Bill Foster in Discover Cayuga Lake. He was on this call. I don't know. I think he's on here still, maybe.
Hey, John.
Yeah, well, yeah.
Hey, every time I'm about to pipe up, my phone rings, and I'm on reservation duty today, so I have to keep muting myself. So I didn't catch the beginning of this conversation, Louise. A couple of there is a lot of monitoring going on at the south end of the lake, and a couple of places I would suspect need additional eyeballs. If you can get up as far as Estes Point or Bolton Point, you know, down when you take the trail down from Black Chin, that is beyond where. Yeah, that is beyond where most of the monitoring is happening at the south end with
So
Bob
maybe
Johnson.
more northerly location. Okay, yeah, I know. I'm thinking I would go to some place that I just like to go to or go to on a regular basis.
Yeah, I think Hogs Hole is going to be an interesting spot too in the southwest corner.
Okay.
Or the Red Cast Park. Yeah.
Okay, yeah, I'll make a few inquiries and see if anybody has a space they suggest. And then go from there. Thank you.
Okay, thanks, Megan. I'm signing off, and you're free to go. I won't ask any more questions.
All right, no worries. Thanks, John. Have a good one.
Yeah, thanks, Megan, for a great presentation.
Thank you. I appreciate it.
Yeah, thank you, Megan. A lot of good information.
Good. Yeah, let me know if you have any additional questions.
Will do. We'll be in touch. Take care.
All right.
Have a
good
good afternoon.
You too. Bye, guys.